Hi there. This is part 4 of the genomics section of the videos. And this part is a starting point to talk about bacterial genomes. In addition to talking about the genomes, I'll start out by talking about the sizes of these organisms, and then a little bit about their metabolic capabilities, which will feed into when we talk about genomics as well. This shows some of the relative sizes of bacteria and some viruses, just to look at some of the complexity, at least based on size. So at the top is oscillatoria and a red blood cell, the size of those compared to an E. coli cell. And E. coli are, of course, free-living organisms. And then into the rickettsia, which is an obligate parasite. And so it is much smaller, has a smaller genome size as well, and there it is compared to a mimivirus and pox viruses and other viruses. The cells on the upper right are in a different scale, and so you can see there's the human red blood cell, bacillus cell, and E. coli, staphylococcus. And then you get down into Haemophilus influenzae mycoplasma, which are parasites, and so they start getting smaller and smaller. And again, there's the mimivirus. On the lower right, at a different scale also, there's a bacterium compared to the mimivirus, chlamydia, which is an obligate parasite bacterium, pox virus, herpes virus, etc. So as cells and viruses get smaller and smaller, they have less and less complexity. And the converse is true also, that as cells get larger and larger, they have, in general, more complexity. And that is complexity of their genomes as well as their structures. So the bacterial genomes and archaeal genomes exhibit this as well. So everything in black up there, those are free-living organisms, usually larger cell sizes. And as you work your way down, then you can see that the ones at the bottom are parasites and symbionts and endosymbionts, and those have smaller genomes and generally smaller cell size. This is a circular phylogram of the tree of life. Bacteria make up most of this phylogram, going from about the 11 o'clock position over to the 8 o'clock position. And then the archaea are there in green on the left-hand side, and the eukaryotes are in pink. So the bacteria and the archaea that I will be discussing in particular, as far as their genomes, are the blue and red circles with the letters in them. This phylogram was produced from the previous one, but it makes clearer the phyla and other groups of organisms that I'll be talking about. So up on top are the Firmicutes or Firmicutes and the Chlamydiae, and then going around the circle all the way to gamma proteobacteria, 4 different ones I'll be talking about there. And then 2 different archaea, one in the Euryarchaeota and one in the Crenarchaeota. Before I talk about the genomes of each one of those organisms, I want to talk a little bit about metabolism. So the most common elements in the cells in bacteria, archaea, and eukaryotes are hydrogen, oxygen, carbon, nitrogen, phosphorus, sulfur, and a few others. Now, the cells must be able to capture and assimilate all of these, but they don't do that all the same way. So in fact, if I gave you hydrogen, oxygen, carbon, nitrogen, phosphorus, and sulfur to eat, you wouldn't live. So you need carbon compounds already built up that you then metabolize into other needed carbon compounds, and for the energy to do those reactions also. Some organisms can use hydrogen as a source of energy and for other purposes, and then others require carbon. Everything requires carbon, but in different ways. They require different types of carbon, including some that can take CO2 and, of course, make carbon compounds out of that. So there are two major metabolic types. Those are autotrophs that can use inorganic CO2 as a carbon source, fix that into other compounds to make their organic compounds needed in their cells. The other types are heterotrophs, such as us, where we cannot fix our own CO2. We have to get carbon compounds already produced and then manipulate those in our various pathways. There are two major types of autotrophs: phototrophs, which use light as an energy source, and those include green sulfur, purple sulfur, cyanobacteria, and algae. And then there are chemoautotrophs that use inorganic compounds as an energy source, and those include iron, sulfur, hydrogen, and nitrifying bacteria, as well as some archaea. There also are two different types of heterotrophs, major groups. One are the photoheterotrophs that use light as an energy source, but they can't fix their own carbon. So these are purple non-sulfur bacteria and green non-sulfur bacteria. And then there are chemoheterotrophs like us, which use organic compounds for their energy sources. So these are most bacteria, the protozoans, fungi, and animals. Because of these major differences in metabolism, you might expect that their genomes would be very different. And they do have some differences, although there are unifying reactions also, and unifying parts of the genome that all organisms have. This is a very general picture of catabolism and anabolism, that is, energy yielding metabolism, and then biosynthetic metabolism. So you start with energy sources, then peel off some usable energy in the forms of ATP, that is phosphorylating ADP, and then you have metabolic products in the end. There is also some heat given off. So in the case of warm-blooded animals, a lot of the energy goes to producing heat, just to make sure that those chemical reactions work properly in those cells. Biosynthetic metabolism begins with these external nutrients, your food essentially. Then the energy from the ATP changes those, for instance, glucose, into other products. Then you get this intracellular precursor pool. More energy is needed for biosynthetic intermediates to produce biosynthetic intermediates, such as amino acids and nucleic acids, and eventually you get to polymeric molecules such as proteins and DNAs that also need ATP, and actually a large number of other reactions as well. So again, thinking of the genome, each one of these different processes requires a set of enzymes, precursor molecules, RNAs, DNAs, amino acids, etc., to produce these compounds necessary for life. And so genes are needed in the genome to produce each one of those different biochemical molecules that will aid in doing all these biochemical processes in the organisms. Building on that last diagram, going into a little more detail, then there are parts of central metabolism, that is glycolysis, the TCA cycle, etc., alternative carbon source production, nucleic acid synthesis, amino acid synthesis, lipid synthesis, cofactor synthesis, transport processes, just a simple process of taking glucose and producing pyruvate, and then sending that into the citric acid cycle, or the TCA cycle, the tricarboxylic acid cycle. That produces NADH, which is necessary for oxidative phosphorylation, another part of the process in the mitochondrion. And in the citric acid cycle, there are several shunts off of that can produce the amino acids necessary for building proteins as well. Again, these are all complex biochemical pathways and cycles, and each step of the way, both enzymes and sometimes cofactors and other molecules are needed to complete these processes, and especially ATP, NAD, NADH, etc. And each one of these processes is really exists within a web or an entire biochemical network, and so they aren't isolated. And this shows that in the middle is glycolysis that feeds into the TCA cycle over here. It also feeds in down here to the fatty acid cycle. There are nucleotides made up here and nucleotides made down here, and then polymers are made in a different process. Many different amino acids, again, are shunts off of the TCA cycle and other parts of the overall metabolic pathways in a cell. Here is yet more detail, and so each one of those lines and dots represent another chemical reaction, biochemical reaction. In the middle, this is glycolysis. Up above that is the pentose phosphate pathway. And then the TCA cycle down here. There are nucleotides made here, other nucleotides made here. This is fatty acid metabolism in here. And this is just sort of an overall view for E. coli. And some cells, some bacterial cells have other pathways that aren't shown here. In the case of humans, you would have other parts of the pathways, and then you would also have some parts of the pathway that are pathways that are missing. And so you actually have to eat those compounds in your food to get them rather than making them in your body. And that's one reason why you're a heterotroph. So E. coli is not a phototroph, but then you have algae, cyanobacteria, and plants, which are phototrophs, and they use the Calvin cycle. So they can take carbon dioxide up here and then attach it to ribulose bisphosphate, which is a 5-membered carbon chain, get initially a 6 carbon chain, which breaks down into 3 carbon chains, and then in doing so they have fixed that carbon to eventually produce sugar, starches, and other compounds as well. So again, this wouldn't be found in most bacteria, but it's found in cyanobacteria and a few other bacterial cell types. In addition to carbon, nitrogen is extremely important. It's present in amino acids and proteins and nucleic acids, so it's vital. So not only the carbon is important, but nitrogen is important. And different bacteria and different organisms perform different functions in the nitrogen cycle. So if you start with atmospheric N2, it's very unreactive. So you need special cells that are nitrogen-fixing bacteria. So those would have certain genes to do that. Then you have other organisms that will take the ammonia produced by those nitrogen-fixing bacteria and make nitrites, and then others will make nitrates, and those can be utilized in terms of what's called assimilation in plants and animals to make their carbon and nitrogen compounds. There also are bacteria that go through the process of denitrification, so they'll change the nitrites and nitrates back into nitrogen gas, N2. Also, as the animals and plants decay or deposit urine and feces and things like that, then there are bacteria and fungi that can take those ammonia compounds and break them down into what's called decomposition, and that will form ammonia, and that can enter the cycle also. And you can see that sometimes if you smell rotting fish, and you can smell that ammonia in there, and that's the ammonification process by bacteria that are degrading the ammonia or the nitrogen compounds in the fish. So you should note that bacteria, any single species of bacterium does not do all of these steps. There are bacteria that do each one of the steps, and so they usually live together and sort of toss these nitrogen compounds back and forth to complete the entire cycle. Another thing that bacteria have to do is they have to build their cell membranes and cell walls, build up their DNA, protein. So there's these polymerization steps to polymerize amino acids into proteins, to polymerize nucleic acids into DNA and RNA, and to polymerize sugars to form polysaccharides. And they also need to make the phospholipid bilayers in the cells to make the membranes. So there are many, many different processes necessary, and many proteins and enzymes needed to perform each one of these functions. And for each one of those pathways or cycles, you need the genes to produce those enzymes and products to complete each one of those biosynthetic systems. Another thing to keep in mind is that there are many, many different environments and habitats throughout the world, so that organisms living in the tropics may have some of the same proteins and genes as those in the polar regions, but those genes need to be slightly different because the proteins need to be slightly different because of the different temperatures in which they're operating. There also are desert regions, there are tropical rainforests, so there are very wet areas and very dry areas, and there are areas that change drastically throughout the year. So the temperate zones and the near Arctic zones change greatly throughout the season. So in each case, the genomes need to include certain alterations in their genomes that allow them to survive in those different, very different conditions. And I should also mention that there are organisms living near thermal vents on the ocean floors, some at very high pressures. And so they have to have proteins that can survive and function under those hot conditions, cold conditions, high pressure conditions as well. And there are organisms that live in clouds, and those have lower pressures, but they're exposed sometimes to more UV irradiation. So in each case, the genomes have to, or have evolved actually, to produce a set of proteins and other products that lead to the survival of those organisms under those varied conditions. So here are just a few examples of those. You have ocean regions, sometimes with volcanoes, sometimes with underwater volcanoes. And so there are a variety of conditions there, both lower pressure near the surface of the sea and high pressure at the bottom of the ocean. You have organisms actually living up to 2 miles and 3 miles in the Earth's crust. And so those also experience high pressures and usually high heat. Then you have organisms that live deep, deep in the ocean, where the temperature in some places hovers around 4 degrees centigrade. And then they can be close to or far away from thermal vents. And so in the thermal vents, the temperature might be 400 degrees centigrade, but then out some distance from the thermal vent, it might be 4 degrees. So there's a wide temperature variation between there. There also are organisms that live high in the atmosphere, and so they undergo different types of pressure, which will form their genome as well. depending on the selection that's going on in those under those different conditions. So here's the first organism I want to talk about. It's Aquifex aeolicus. It's a very ancient bacterium living in hot pools, and it has a rather small genome. And so let's talk about that one first. Here it is. Here you'll notice it has flagella, so it can move within the hot pools. These were isolated from hot pools in Yellowstone Park as well as in New Zealand. They're able to move, so they can move through the pool to both to find nutrients and to alter the temperature, to find a temperature that's optimal for them. Again, very ancient organism, probably originally evolved early on Earth in very hot water. So here's the genome, 1.5 million base pairs, which is relatively small for a free-living bacterial genome. Remember when I was talking about free-living organisms, the cutoff's about 1.2 to 1.3 million base pairs between a free-living organism and a parasite or a symbiont. 1,522 genes, 1,778 genes were reported in a different study. 6 to 7 ribosomal RNA operons, and so this is enough so that they can have a doubling time as rapid as every 20 to 30 minutes, and 44 tRNA genes. So these are among the oldest extant bacterial lineages. They've been around for billions of years. They're thermophilic, found near underwater volcanoes as well as hot pools. Their proteins are thermostable, so their DNA polymerase, their RNA polymerase, all of their enzymes are thermostable. Part of their stability comes from the fact that they have higher concentrations of cysteine in their proteins, and they have disulfide bridges in the proteins to hold them together, so they're even in hot temperatures they don't kind of fly apart. They're aerobic, although some can live under low oxygen conditions by reducing sulfur instead of oxygen to form hydrogen sulfide. So it reduces oxygen to form water, that is H2O. Approximately 16% of its genome appears to have come from a member of the archaea. So for billions of years, this species has been living close to archaeal species. And so over that amount of time, through horizontal gene transfer, then it's picked up many genes that worked within its genome. That is, that they were necessary for the survival of this organism. Now, they all live near underwater volcanoes, so this is probably where those gene exchanges happened. It's a chemolithotroph, that is, it fixes carbon from CO2, but it's not photosynthetic, so it has those genes for fixing carbon from CO2, a Calvin cycle. The enzymes used in aerobic respiration are very similar to the enzymes in anaerobic bacteria. So it may have been originally 3 billion years ago anaerobic, and slowly the genes, at least some of the copies of the genes, changed so that they could be utilized for aerobic respiration as well as the oxygen concentration in the atmosphere and in the ocean increased. This is a diagram of the Aquifex aeolicus genome. On the circle, you can see in orange, those are genes that are on one of the two strands of the DNA, and the yellow on the outside, those are genes on the other strand of the DNA. The red arrows inside, those are the tRNA genes, and then the blue arrows inside of that, those are the ribosomal RNA genes. So you can see they're all pretty much separated. Also indicated on the outside of the circle are transcription factors, sigma factors, and response regulators. Because this lives in hot areas and it needs to control the temperature where it grows, so it's moving around, it has different ways to control which genes are turned on and which genes are turned off. So it has quite a few transcription factors. It has response regulators, again, to react to different environmental conditions. And then the sigma factors. Sigma factors are unique to bacteria, but they're similar to transcription factors in that they regulate transcription of the neighboring genes. So one thing you can see from this picture of the genome is that the genes are very close together, concentrated together, although the tRNA genes and the ribosomal RNA genes, are scattered throughout the genome. The next genome I want to discuss is E. coli K12. So this is one of the strains of E. coli. And remember that all E. coli are not equivalent. So this is one that exists in human colons, and it's actually a minor component of the the microbiota of the colon. So remember, I have shown this one before where K12 is in the middle there, along with W3110, another strain. It's very different from the 0157 strains, which are deadly, can be deadly, and those are closer to Shigella dysentery. And it's also related to other Shigellas, which are can be normal bacteria in some organisms, but certainly not in humans. So a lot of different strains of E. coli. This is just one of them. On the top left, those are E. coli cells grown in culture, and you can see that some are dividing lengthwise. This is colorized, so they're not really red. There's an E. coli on the top right, just showing some of the major features, including the flagella, where the DNA is, the nucleoid, ribosomes, etc. On a plate, it would look similar to this, depending on what type of growth media you're growing on. It does vary a little bit depending on the growth medium, but you can see the individual colonies. And it also can be found on fruits and vegetables, so you're probably eating it all the time. K12 is fine, but if you get 0157, then you can have severe diarrhea caused by that severe disease. So because you can find it in many different places, that is, it will grow in human colons, which is an anaerobic environment at about 37 degrees centigrade, and it can grow out in a plate, and you can grow it at 37 in an incubator in an aerobic environment on certain media. So it needs a larger genome in order to react and to express the proper genes for the proper environment. Otherwise, it will not survive. The previous bacterium, Aquifex aeolicus, pretty much has a stable environment that it lives in. So it requires fewer genes. It's not adjusting to many different conditions. But E. coli is able to adjust to many, many different conditions. So here's some information on its genome. It has a genome size of about 4.7 million base pairs, 4,289 genes, including 7 ribosomal RNA operons, which we talked about quite a while ago. 7 ribosomal RNA genes allows this organism to produce enough ribosomes to multiply or double in about 20 minutes. And 86 tRNA genes. So a lot of tRNA genes, more than it actually needs, but then on the other hand, it can choose which ones to express at what time to make the proteins that it needs. So it's a facultative anaerobe, that is, it grows in under anaerobic conditions in the colon, but it can grow outside in oxygen as well, so it can be aerobic. It's commonly found in the lower intestines of warm-blooded organisms, and it colonizes infants within 40 hours of birth. and that's one reason why the birth canal is so close to the anus, in that the infants are colonized very early, not only from birth, but also from drinking milk from breast milk. Optimal growth is 37 degrees centigrade. As I've already said, it's related to Shigella, and it lives outside of the host for a relatively short time, but it can actually live there for hours to days, depending on where it is. It can live on a wide variety of substrates, which also is indicative of the large genome. It can adjust to many different carbon sources, nitrogen sources, etc. So it can ferment sugar to produce lactate, succinate, ethanol, acetate, carbon dioxide, and a small amount of hydrogen gas. So that tells you something about the number of enzymes it needs to do those different processes. It can also oxidize pyruvic acid, formic acid, hydrogen, and amino acids, and reduce oxygen, nitrate, dimethyl sulfoxide, trimethylamine, and oxide. And it has flagella, so it's modal. It needs all of those genes, and we've talked about how many genes are needed to make flagella. It's quite a number. And then conjugation. It can transfer DNA from one E. coli cell to another. So there's another system that it needs genes for. And it diverged from Salmonella about 100 million years ago. And that's coincident with the separation between reptiles and birds and mammals. And if you look at reptiles and birds, they're more used to having Salmonella in their intestines and not E. coli, and we're just the opposite of that. So part of the genome also has some relationship to the microbiome of those organisms. What genes and gene products are compatible with those different microbes that live on and in those organisms. Here is a diagram of the genome of E. coli K12. That has 4,639,221 base pairs. You have an origin of replication on one end of one side of the molecule, and a terminus of replication on the other side. So there's replicor 1 and replicor 2 there. The yellow and orange blocks there indicate the genes on one strand versus the other strand. The next line in, those are little green arrows. Those are all the tRNA genes in the E. coli genome, and the red arrows inside of that are the ribosomal RNA gene loci for each one of those, and there are 7 of those. The internal sort of sun-like, corona-like diagram in the center of those yellow markings indicate AT and GC-rich blocks, but I think you don't have to pay any attention to those at the moment. Genomes can be subdivided into functional groups just to indicate what sorts of gene functions are present in those genomes. So at the top is the functional class, and then the number of genes in that class and the percent of the genome that those make up. So you can go look at regulatory genes, 178 genes in that group, which makes up about 4% of the genome. And then going down all the way to other processes means those haven't been completely categorized. And 38% of the genome has unknown functions, and that always surprised me, that a lot of genes, even in E. coli, when they were identified, no one has any idea what they're doing. So this is an organism that's been studied for decades genetically, and so you would think that people had pretty well dissected the whole genome of this organism, but when they sequenced it, they found that this wasn't true, that there were a large number of genes in E. coli that were completely unknown, unexpected, and many had no known function, and many still have no known function. The next group that was somewhat of a surprise is that there were 87 genes, about 2% of the genome was made up of viruses or phage, transposons, and plasmids. So a large proportion of the genome, well, a relatively large proportion of the genome, was made up of viruses and transposons. And this holds true in most genomes that I'm aware of, including the human genome, where up to about 8% of the genome is made up of virus sequences, and about 40% consists of transposons and retrotransposons. 4% of the genome consists of regulatory genes and proteins, so that these are the ones that are controlling the expression of other genes. And so in something like E. coli, where you have to switch from growing anaerobically to growing aerobically to growing on this in this environment, in that environment, that's expected. That 178 genes are regulatory genes. That is, they encode proteins that control the expression of other genes around the cell, around the genome, and around the cell. About 16% of the genome consists of genes that produce proteins necessary for cell structure, membrane proteins, structural proteins, and transport and binding proteins. So these are really forming the structure and the communication within the cell and from within the cell to outside of the cell. So a fairly high proportion, and this holds true of many different genomes. A large number of genes are necessary for these functions of making the cell and then communicating, transporting things in and out of the cell and around the cell, within the cell. About 13% of the genome encodes proteins necessary for building of different compounds in the cell, including those that produce DNA, RNA, proteins, and fatty acids. So these are the major biosynthetic pathways for producing the things necessary in the cells, as well as for replicating those cells also. Here's a pie chart showing the different functional groups of genes within the genome of E. coli. Down on the lower left, you can see in this particular analysis, about 50% were classified and 50% were unclassified. The largest category is in energy metabolism. That would be glycolysis, the TCA cycle, ATP synthesis, etc. About 780 genes are involved in making the proteins, the enzymes for that. About 10% for DNA replication and nucleic acid synthesis, about 10% for transcription, and then about 10% for translation, also both in translation and amino acid synthesis. There's also 14.8% in transport and binding, that is, moving things around in that cell to where they're needed. So this is a fairly complex cell than E. coli. And then other functions such as toxin production and others. So this is for E. coli, a microscopic organism of some complexity, and you can see how complex it really is. It's got a 4.7 million base pair genome, encoding 4,289 to 4,300 genes, making various proteins for that cell in order to function. And so, although it is a microscopic organism, it's already fairly complex with a fairly decent size genome for a bacterium. So that's the end of the first part of bacterial genomes. Next time I'll talk about other bacterial genomes, as well as a couple of archaeal genomes. So thank you. Talk to you later.